So I want to open questions up to the audience, and I also want the speakers to be able to ask questions to each other. But I thought it would be a great idea to just kick it off with responses to the final question of the last talk, right? How useful is it to collect all of this data? If we collect it, how useful is it to share it? How do we manage this enormous data problem? Do we want to share the raw data? Do we want to share just the processed data? Do we want to share compressed versions of the data? Have you explored possibilities in any of these domains? I'm really curious to get the thoughts across this kind of diverse group of people leading different angles of neurophysiology technology development.
Same answer twice as a unit identifier. I don't know whether or not that's a too rigorous. The problem is it cuts your unit rate way down. People want lots of units. Maybe those units are fiction. What do we do? I mean, there's a question. I don't have an answer.
I don't have an answer either. It's the same problem in the voltage imaging world that the shape of the data is a little bit different because it's a lot more pixels but a somewhat lower data rate. So it's a kilohertz, but there's 400,000 to 1 million pixels per. But that, as Tim said, the actual information, if it's 300 cells and it's at a kilohertz, that's very manageable. To store 300 waveforms sampled at a kilohertz, but we also don't trust the signal extraction, and there's background issues and blood flow and so on. And every time we've tried to reduce it, then as we start to do more and more spike-triggered averages and look at subtle correlations, we see correlations, and then we wonder, well, is that really there, or is that some noise being driven by some common source of noise, and we can go back to the original data. And so finding what's the intermediate level of compression, maybe there are things that where you can compress, maybe not calling all the spikes, but storing the spikelet formed in some lower dimensional way and keeping some of the information enough so that you can go back if you don't trust the final outcome, but you're not keeping all of the noise that's required and during all the silent times. I think there might be intermediate approaches, but I haven't.
my view of how I think about that with respect to, a shaft-based sampling. I mean, I was trained as an optical chemist. The first thing you do and last thing you learn is remember you're just sampling. You don't have all the information. And so when we generate data, when my community generates data, we don't have anatomical insight. It's one of the advantages of the imaging. We have at least some anatomical insight. I mean, I love these pictures that Adam was making for propagation. Even if it is a slice, it's still, amazing insight as to how things behave. I think electrophysiology just uses all of that information, and I think that makes it much tougher to model. We're trying, but I think we're a long way from sort of definitive analytical understanding. The first thing that people that often forget is that neurons on point sources. They're distributed discharge devices, so you get weird waveforms. You don't know where your sensor is with respect to this non-point source. Brains are complicated.
I mean, I think that the modeling, I mean, that's the sport. That's what we're trying to do after taking the data, but that's still very much an artisanal task. And who knows, if you pick the wrong, I mean, trying to figure out which models better explain the data is sort of what we're trying to do. But that's a different problem that doesn't scale really, I think, to the problem of everybody's taking lots of data and you want to be able to share it and store it and test it against lots of different models.
I saw Jerome's hand first.
Yeah, so the spike waveform does change depending upon where you are in the cell and the not only just as a function of position, but also as a function of the history of the cell. At a given spot in the cell, the waveform depends on what the cell was doing recently. And so those maps showing the back propagation are for isolated spikes against a quiet background of the cell not doing much in a slice, and we're surfing on the sort of 50% point of the upstroke. And so we're ignoring all of those details which you just raised about the spike waveform. If you care about the spike waveform, then you have to look at the data in more detail and look at the actual waveforms, and you do see that there are variations. So that was just to visualize sort of the wavefront propagation. Yeah. But I think it's also important for the electrode-based reporting that two spikes from the same cell reported from different electrodes could look very different, and could depend on the history of the cell.
Yeah, I don't want to monopolize things here, but these high-resolution devices that we've been using over the last couple of years, what we see is there is variation in the spike waveform, but there's little variation in the spatial extent of that discharge. It's difficult because you have to agree that you can recognize accurately every spike, not its average waveform, and then pick it out and then fit each of those individual spike waveforms and ask them. But from the standpoint of spatial sampling, that pretty close is the answer to how do they vary spatially. The answer is not much.
Yes, well, this is definitely the direction that we're taking. What we've seen here is actually in two trials, we could decode this type of activity, even for individual fingers, a small number of trials will allow us to really decode which area corresponds to which index that's being moved. And so the hope is that the next set of experiment would be a plug-and-play BCI in the intraoperative room. And then a few years later, we'd have the opportunity to do this over extended durations of time in our epilepsy clinical trial. So we're heading in that direction right now.
Wow. Went from 20 or neurons being a good day to I want 1,000. But there are what 10 to the 13 or something neurons down there. I think that it's still a question of how much you can understand about. What question you get to ask, given such a tiny sample of a large object. Doris's data convinces me that there are places where it really works. And then there are other places where it might not work as well. I think that's a case where it turns out that face recognition is remarkably localized. I don't know how many, I'm not practicing neuroscience, I'm a gadget builder. So I don't know how many places for which that is an analogous insight. But what Alipasha, how many voxels are you interrogating in an average experiment?
The voxel acquisition rate is 160 megahertz.
Okay. I see. So you can just decide what time resolution you want based on that voxel acquisition
What
rate.
time resolution, what space, scale, they are all interchangeable.
I worry at that rate you're just running out of photons.
Well, we are having, when you do the estimation, so we are having about 100 photons or so per point. That's about.
So for voltage imaging, one of the limitations is shot noise. And typically we're looking at we want a sensitivity of about 1% in the fluorescence value at a kilohertz bandwidth. So to have 1%, you need 10,000 photons per signal source per millisecond. So that's 10 to the 7 photons detected per source per second. And then depending upon the brightness of your sample, and that tells you how much laser power you need to put into the tissue. As you put in more laser power, you either bump into heating and photo damage issues. You can't put in more than about 200 milliwatts into the tissue, so that's on one wall that you want to steer clear of. There's also limits on the time average intensity. You don't want to get too much focal heating, and then photo bleaching and so on. The other problem you run into is as you try to increase your field of view or increase the number of sources you're recording from, most of the light that goes into the sample, of course, passes right through the thing you care about and goes deeper into the sample and rattles around and contributes to background. And so the total background level is proportional to the number of things you're trying to record from, but that background is shared across basically all of your sources because it's so broadly distributed. And so your signal to background ratio goes down as you try to image more samples, particularly if they're densely spaced. And that eventually contributes to shot noise, to decreasing your signal. Another noise source, which is subtle and I hadn't anticipated, is that the sub-threshold voltages of many neurons are much more strongly correlated than the spiking patterns. And so if you have out-of-focus cells, their sub-threshold signals will be at least partially correlated with the sub-threshold signals of the cells you care about. And that makes it hard to use activity-based segmentation algorithms to separate those two, because signal and background are correlated. You don't know how to apportion your photons to the cell you're interested in versus the background. And so one way of addressing that is we use the spikes to try to find the footprints of the cells. And then, so we sort of high-pass filter, use the spikes, cross-correlate those with the images to find the footprints, and then try to use those spatial footprints to separate out the genuine sub-threshold signal of the cell we care about versus all the out-of-focus signal.
So maybe so maybe I can say something about that. So our motivation was in this regard twofold. One was, of course, by trying to look at this scaling of information representation to see what, to learn something more fundamental about how information is represented or processed within the brain, maybe learning about some of the computations. But it was, the other thing was also that we felt that this would be also very informative in terms of being able to put our efforts to the locations or the spatiotemporal scale, etc., that would maximize the extraction of information. So that was the motivation. I would expect that at some point, obviously, the information would be saturating. It is not the infinite system, but I think a lot of that, our sense is that it will depend a lot on the behavioral context as well. So it becomes very quickly a question of what is it that you want to understand. For instance, we see that the scaling that I showed you can be very different when the animal is just in darkness and we are recording spontaneous behavior versus when the animal is engaged in a task. So the dimensionality is reduced when the animal is engaged in specific tasks, where it depends on the complexity of the task and so on. So that's why I don't think that is at this point, or I cannot say at this point, how can we really decide on what the right scale and amount is that we want to record. Ideally, you would be interested in a technology that can allow you to record from maybe these reduced dimensions or these latent dimensions, but that doesn't map one-to-one to neurons. So that's the difficulty.
Yeah, I think that a question again relative to that is that the paper of these high-density probes is up on bioarchive, so you should have a look if you're interested. But among the things that we saw were apparently very small sources of discharge that you mostly wouldn't see with a low resolution device. And the question is, should I care? And so that our opinion is they mostly generate units that are unreliable unless we correlate them with a receptive field, in which case you see a bunch of things that you wouldn't have trusted if it didn't have such a clear receptive field. I honestly don't know how I should think about that. But so clearly, there was information that was sort of marginal until you had a secondary channel of confirmation, in which case it became looking pretty reliable. What does that tell you about how to conduct a research project, rather than just monitor behavior and record spikes? It was a surprising observation to me, because I would have thought that data wasn't very useful.
Along the same lines, I think we also found that reporting from core neurons, because these correlations are weak and so distributed enough, it will allow us also to make significant statements about the expected outcome decision making.
like to follow up with a comment here. I'm not sure actually that we should be studying connectivity based on in the spike band, right? So there's a lot of literature that's published recently where if we look at lower frequency bands, we look at interacting nodes across broad brain regions. So I'm wondering if we start analyzing the data in these frequency bands, and then where we find interacting networks, focus on data analysis with individual units only in these nodes, and then the dimensionality would be reduced, and data probably would be able to handle it.
I'll just comment, since I was the original cause of that a lot. No, this literally caused a panic at the Brain Initiative. We had a follow-up phone call with all of the project managers and John Nye saying, 2.8 an hour, what do we got? You can't pay for that. And so I think that some compromise about, what we're going to post and how we're going to post it, I think that making it all public is absurdly expensive and inefficient. And so I think you can only describe, brands of data, and if somebody wants to send you a box of hard drives, you can copy it off and send it back. But I think genuine online sharing just doesn't scale the way the data sets are scaling.
Sorry, maybe one point along these lines, because we have been also thinking about that a little bit, discussing within the group. Is that I agree with your point, Sam, where you say that if we can compress the individual units, then the amount of data would be tremendously lower. The other thing we have been discussing is a lot of these metadata is what is needed in order to be able to, in a meaningful manner, being able to make sense of the data. And without that, it's just doesn't make any sense. As you said, behavior information, etc., is very important. And what we have been discussing is basically whether in some cases where we have a lot of stereotypical information and details about how the data is reported, and we have confidence, whether we can just describe the data and then re-report the data if needed. We are spending maybe 2 days recording data, and then, as you were pointing out, half a year analyzing it. So that could be maybe a more efficient way, especially with distribution and dissemination of the technologies that are supposed to become more widespread use and cheaper and so on. I just wondering whether this could be also a viable approach of having recipes on how to recreate the data instead of having.
Yeah, I agree. I mean, I think that the sharing of these massive movies and the notion that we're going to share everything we record, not only is it very expensive, but it will lead to a sea of basically meaningless data, of noise, and it will make it very hard for people to actually use or find any of these data. And as Alipasha said, what they're paying us for really is the analysis of the data and the interpretation. And so we've interpreted these guidelines as saying, we'll share the results of the analysis and the distillation that goes into papers, or that goes, or maybe one step upstream of that. But it's hard to imagine a scenario where the raw videos are actually useful